In the last video, we talked about events that happen before the first week of development. Now, in this video, we'll talk about events that happen in the first week of development. The first week of development includes ovulation to implantation. On the 14th day of ovarian cycle, due to rupture of the Graafian follicle under the influence of the LH cells, secondary oocytes are released into the abdominal cavity surrounded by cumulus ophorus. This process is known as ovulation. After the discharge of the ovum into the abdominal cavity, here in this animation we can see that after the ovulation the ovum is cast by the fimbria and then it is transported to the most dilated part of the fallopian tube, which is known as ampulla, where the fertilization takes place. And this movement is facilitated by the ciliary movement of the lining epithelium of the fallopian tube. These are some of the basic parts of the uterus, nothing to mention. You can see in the figure and note it down. Now let's come to the main thing, that is fertilization. In simple words, fertilization is the fusion of male and female gamete to form zygote. And since the ampulla is the most dilated part of the uterine tube, so the fertilization takes place here. The lifespan of the sperm is 3 to 5 days, but the life of the ovum is 12 to 24 hours. It means the fertilization needs to happen within 12 to 24 hours of the ovulation. Both the sperm and ovum are haploid in number. It means both of them carry half number of chromosomes, that is 22 autosomes and 1 sex chromosome. But the zygote is diploid in number. It means it contains a whole set of chromosomes, that is 46 number of chromosomes. Among them, 44 is autosomes and 1 pair of sex chromosomes. The sex chromosomes can be XX, which denotes female, or it can be XY, which denotes male. So in simple words, the sex of the fetus is determined by the biological father of the fetus. About 300 million sperms are ejaculated. Among them, only one of the sperm fertilizes the ovum. Sperms move in the female reproductive tract by the ciliary movement of the sperm and by the contraction of the uterine muscle. Before the process of fertilization, sperm goes under capacitation and acrosomal reaction. And these help sperm to cross three barriers, that are corona radiata, zona pellucida and oocyte membrane. These are the borders, or these are the barriers that a sperm need to cross before fertilizing the ovum. Now, what is capacitation? Capacitation is the process in which when the sperm passes through the female genital tract, it gets conditioned, which means sperm increases its capacity of penetrating corona radiata, which helps in fertilizing the ovum. Some extra information is given here, like during the process of the capacitation, the layer of glycoprotein and seminal plasma protein is removed from the acrosomal region of the head of the sperm. Now let's come to acrosomal reaction. When the acrosomal head of the sperm or the acrosome part of the head of the sperm comes in contact with the zona pellucida, a reaction occurs due to which acrosine and trypsin is released, which helps in the penetration of the zona pellucida and fertilize the ovum. Now let's see the three barriers of the secondary oocyte. As we know, a ovum after being released, it contains secondary oocyte which is covered by cumulus ophorus. Cumulus ophorus further rearranges itself to form corona radiata. Now the first barrier is penetration of the corona radiata. Only the capacited sperm can penetrate corona radiata. Now let's come to the second barrier, which is penetration of the zona pellucida. Zona pellucida is a glycoprotein coat secreted by the primary oocyte and granulosa cell. Penetration of the zona pellucida occurs when the sperm comes in contact with the zona pellucida and acrosomal reaction occurs, due to which acrosine and trypsin-like substance are released, which helps in the penetration of this barrier. Now, there is a special thing that occurs after the penetration of the zona pellucida layer. Since we know only one sperm can fertilize the ovum, so after the crossing of the zona pellucida, spermatozoa comes in the close contact of the oocyte membrane, which releases lysosomal enzyme, due to which a conformational changes occur in the zona pellucida, and after which the entrance of the one sperm causes other sperms not to be able to penetrate the zona pellucida layer, which means one sperm after entering the zona pellucida prevents the entry of the other sperm, which helps to prevent polyspermia. Now let's come to the third and last barrier, that is oocyte membrane. Simply, it is the fusion of the oocyte membrane and the spermatozoa membrane. After crossing of the oocyte membrane, only the head and neck and the middle piece of the sperm enters the oocyte membrane, and the tail part called spermine and the cell membrane of the sperm remains outside. Now, after finally crossing all these barriers, sperm finally fertilizes the ovum and the secondary oocyte finally completes its second meiotic division and forms zygote. Restoration of the diploid number of chromosome occurs and metabolic activation of the secondary oocyte occurs. Now, let's have a look at all those processes we studied till now in this small animation. At the very first, we know On the 14th day of the ovulatory cycle, ovum is released into the abdominal cavity by the process called ovulation. Then the ovum is cast by the fimbriae, and then via infundibulum it goes into the uterine tube, where it travels to the most dilated part of the uterine tube, which is called ampulla. Meanwhile, during the intercourse, the sperm ejaculated in the vagina or in the female genital tract approaches towards the uterus. At first, the sperm reaches the external os and enters into the cervix. Then, the sperm enters into the uterine cavity via internal os. Here, the movement of the sperm is facilitated by the ciliary movement of the sperm itself and the muscular contraction of the uterine walls. Sperm, after reaching the uterine cavity, approaches towards the intramural part and enters into the fallopian tube. And these are the site where the sperm gets conditioned, which we know helps in the capacitation reaction. Sperm, after entering into the uterine tube, now slowly approaches towards the most dilated part of the uterine tube, which is called ampulla, where the ovum has already traveled and is waiting for the sperm to fertilize it. Now let's look at the process of fertilization. As we know, before the fertilization, sperm has to cross 3 barriers. So here we can see the capacited sperm crosses the corona radiata barrier. After the crossing of the corona radiata, then comes zona pellucida. At first, when the acrosomal head of the sperm comes in contact with the zona pellucida, the acrosomal reaction occurs and we know acrosine and tyrosine release, which then helps to cross the zona pellucida barrier. Now, after crossing the zona pellucida barrier, we know sperm comes into the contact of the oocyte membrane and lysosomal enzyme is released, which causes conformational change into the zona pellucida, which then do not allow other sperm to enter the barrier. Finally, the last barrier remains, which is called oocyte membrane. So, the membrane of the sperm comes into the contact of the membrane of the oocyte and fusion reaction occurs. The sperm leaves its tail and cell membrane behind and the remaining part of the sperm fertilizes the secondary oocyte. Finally, the fertilization occurs and zygote is formed. After the formation of the zygote, cell division begins and cleavage starts to form. At first, the zygote divides to form 2 cell stages. Then 2 cell stage forms 4 cell stage. Then 8 cell stage. And then 16 cell stage. 16 cell stage is also called morula. Now, what is this cleavage? It is the series of mitotic division by which zygote divides and forms smaller number of cells. They are called blastomers. Here, the 16th cell stage is also called morula or mulberry stage. Morula stage enters into the uterine cavity where the fluid from the uterus, which is also called uterine milk, enters inside and the inner cell mass moves to one side and the outer cell mass forms a ring around it. After the entry of the fluid, a blastocele cavity is formed and the zona pellucida disappears, and now this stage is called blastocyst. Now once again, let's see what happens to the morula inside the uterus in this small animation. Morula, after being formed into the ampullary region, now it travels back to the uterine cavity where the fluid from the uterine cavity, which is also called uterine milk, enters inside and then the inner cell mass moves to one side and the outer cell mass forms a ring around it and a blastocele cavity is formed and it forms blastocyst, which is then embedded into the endometrium from where the development of the fetus begins. Now let's see how blastocyst forms. Here in this figure of morula, we can see after the entry of the uterine milk, we can see how the inner cell mass moves to one side and forms these clusters of cell represented by blue color, and the outer cell mass forms a ring around it, which is denoted by pink color, and the space in the middle, that is called blastocele cavity. Blastocyst is formed after 5 to 6 days of the fertilization in the uterine cavity. It is the stage which gets implanted within the endometrium of the uterine cavity. The inner cell mass is called embryoblast and the outer cell mass forms the future placenta. Now let's see what are the functions of the zona pellucida. Zona pellucida acts as the barrier for the oocyte. It facilitates the acrosomal reaction. It prevents polyspermy and it prevents abnormal implantation of the blastocyst. Now, what are the results of fertilization? If we see serially, then at first, completion of the second meiotic division of the secondary oocyte occurs. Then in second, formation of the zygote occurs. Third, restoration of the diploid number of chromosome occurs. Fourth, determination of the sex of the new individual. And fifth, initiation of the cleavage begins. Now comes the last event of the first week of development, that is implantation. Implantation. It is the process of embedding of the blastocyst into the uterine mucosa between the opening of the two glands on the anterior or posterior wall of the body of the uterus close to the fundus. Embedding occurs within the functional layer of the endometrium. As we know, uterine wall has three layers: perimetrium, myometrium, and endometrium. Endometrium further has three more layers: basal layers, compact layer, and spongy layers. Here, the compact layer and the spongy layer is together called functional layer, where the embedding of the blastocyst occurs, and shading of this layer also occurs during the menstruation. Apart from the normal implantation, there are some sites of abnormal implantation. It is a condition where the implantation of the blastocyst occurs apart from the site of its actual implantation. Abnormal implantation leads to abnormal pregnancy, which is also called ectopic pregnancy. Now, what are the sites of abnormal implantation? It can be anywhere into the uterine tube, like ampulla, isthmus, intramural part, and infundibulum, or ovaries, abdominal cavity, or internal os. Among all these sites, the implantation at the isthmus is the most dangerous site of implantation because the fallopian tube can get ruptured here. Here you can see the normal site of the implantation and the abnormal site of the implantation. Here you can see that the blastocyst gets implanted into the uterine mucosa in the normal site, that is in the anterior or posterior wall of the uterine mucosa close to the fundus between the opening of the two glands. It is denoted by green color here. Now, these are some abnormal sites of the implantation, which is in the internal os, ovaries, abdominal cavity, ampulla, and the fallopian tube. This is all about the first week of development required by a first year medical student for their written and viva.